Okay. Can you see my screen?
Yes, sir.
So again,
Yeah.
check left hand corner, left,
Yes,
Yes.
sir.
yes, top,
Yes, sir.
Yes.
right,
Yes, sir.
and bottom.
Yes.
Okay. Just a second. I'm getting a call here. Hello. Yeah. Okay. ठीक है ठीक है. Okay. Hello. Hello. Sorry for that. Huh?
Yes, sir.
Hi, sir. So, if you remember last time, we had talked about uh, the difference in what happens after the RNA is made by RNA polymerase. Okay, with respect to prokaryotes and eukaryotes, and we said that in eukaryotes, after RNA polymerase makes RNA, uh, uh, what you also have is a modification that occurs to the RNA which is actually synthesized, and there are several modifications that occur which are referred to as post-transcriptional modifications. And we started looking at some of them, and the first we looked at was splicing, where we said that as part of splicing, after the RNA is made, a lot of pieces of RNA are actually removed, or what is called as spliced out. Okay, and these spliced out, what are actually speaking non-coding regions, are referred to as intervening sequences or introns, and those are removed. The remaining DNA, remaining RNA, which is referred to as exons, are joined together. Okay. For example, as I said, if this is a DNA and then this is RNA which is freshly made by RNA polymerase, then some of the sequences shown here in colors, green, blue, red, and yellow, and this light blue, are actually sequences which code for the protein. So they are what are called express codons. So they are kept, and all the sequences which are in between them, which are referred to as introns, are removed as part of the processing, which is called as splicing. Okay. The second modification I said was 5' capping, and here I said there's an unusual reaction that occurs at the 5' end of the RNA, and this reaction involves forming what is called as a GTP cap. Okay, or I should say not even GTP, a guanosine cap is more correct. Okay, and what you find is that a guanosine moiety is added to the 5' end via a very unusual triphosphate bridge, which is called the 5' triphosphate bridge, because it connects the 5' carbon of two nucleotides. Okay, one which is part of the RNA, which is the 5' end, and the other which is of the guanine, which is actually the cap. Not only does this phosphate formation occur, in addition, there is also methylation which occurs at the level of the guanine and a couple of residues which are part of the RNA. Okay, shown also in this fashion. So this is the RNA that we are talking about. This is a 5' end, and now there is this phosphate. Phosphate 1, phosphate 2, phosphate 3, again linking to the 5' of a guanine or a guanosine, which is methylated. In addition to this methyl group, notice the methyl groups also on the RNA. So the second modification is what is called as 5' capping and methylation, okay, at the 5' end. The third modification we talked about was poly-A tailing, okay, and we said that a poly-A tail is added to the end of the RNA, and this is added in a non-template requiring fashion by a polymerase. Okay, and because it is a non-template type of addition, many times there is variability in how many A's are added at the end, and you can have anywhere from 40 to 200 A's which make up the tail. Okay, and this is also shown here. RNA is made, capping is done, splicing also occurs, and the RNA comes out, and the poly A polymerase adds a whole bunch of A's. Okay, and we stopped here and we said that the next modification is what is called as RNA editing. Okay, and this is also very interesting. Okay, and it's very unique in that in almost all the living organisms, this is one of the evolutionarily conserved properties of RNAs. Okay, so what is RNA editing? In terms of defining it or explaining it, it's a process or it's a molecular process through which some cells, okay, then this is very important, some cells can make discrete changes to the specific nucleotide sequences within an RNA after it has been generated by DNA polymerase. Okay, so this is very interesting. This is almost like causing a mutation. Okay, so think of this, a molecular process through which some cells can make discrete changes to specific nucleotide sequences. Okay, and one very nice example which is given of this RNA editing is this scenario here. Okay, now this is an example of a gene which is read to make an RNA. Okay, and once this RNA is made, interestingly, what happens is in the liver, processing that occurs is different than the processing which occurs in the intestine. Okay, it's the same gene, it's the same mRNA, and see what happens in the liver. It gets translated, and you get a protein which is called as ApoB100. Okay, now the same RNA in the intestine is RNA edited. Okay, and there is a little sequence which is a CAA sequence at this location here shown here, and that is edited such that the C is converted to a U. Okay, the minute that happens, this becomes a stop codon. So in the intestine, the same mRNA is read, but it is read only up to this point, and you get a shorter protein. Okay, and this shorter protein is called as ApoB48. Okay, now this type of editing, as it is called, where in this cell a specific change is occurring at the molecular level, where a C in the RNA is being converted to a U. Okay, and this how is this done? By oxidative deamination. Okay, and such phenomena where RNA is edited to make a different protein is referred to as RNA editing. Okay. Now, in addition to this, there are a lot of other things that can also happen depending on the biological system we are talking about. Okay, and these are some very interesting things. Okay, one which is called as alternative splicing. Okay, now see what this means. Okay, now let us say this is the pre-mRNA, and these colored boxes that you have are actually the exons. Okay, so what can sometimes happen in biosystems is that these exons are joined together in two different ways. Okay, so in one way, red and yellow and blue are joined together, everything else is removed. Okay, and that is shown here. See this red going up, down to yellow, which is going to the blue. Okay, and that gives you this RNA. In the other case, what happens is red is then connected to green, which is connected to blue. So you get this RNA. Okay, so see what has happened. We have seen what is called as mRNAs which are spliced alternatively, where now it is not only removal of introns, but in some cases exons are also what you call as swapped, and you end up getting two different proteins from the same pre-mRNA. Okay, and this is different from this. Here it's a very specific single nucleotide change, which is therefore referred to as RNA editing. Here it's a more gross change where different pieces are joined together. Okay, now such type of what you call as phenomena can also occur in many different ways. Okay, and these are some ways where people know that this happens. Okay, so this first is called as one type of splicing, what is called as constitutive splicing. That means this is what happens so normally. Okay, so you have yellow, pink, blue, and green exons. Intermediate are all the introns. So So these are removed and these are joined together. The joining is indicated by this arrows here. Okay, so you get this RNA which has got all the 4 exons. Okay, now see what is happening. Here I am skipping an exon. Okay, so this is called as alternative splicing of a type. So now yellow is connected to blue to green. So I get another RNA, another protein. You now see what happens here. In this case, what has happened? I have taken this exon and I have taken a part of this. Okay, and then fully this and fully green. So now I have another different protein. This type of is called intron retention. Okay. Now see, mutually exclusive exons. Okay, so this is what I showed last time. So in one case it is yellow, pink, green. Other case is yellow, blue, and green. Okay. Then you have alternative 5' splice site. Okay. Now see what happens. Here I join in such a fashion that I retain this yellow and part of this pink and then blue and then green. Okay, and look at in this case I have taken yellow, pink, part of blue and then part of green and then joined together. Okay, now basically what we are talking is alternative 5' splice site and 3' splice site. Okay, so what do you mean alternative 5' splice site? Normally this should be splicing here, but it ends up splicing here. Okay, that's why it is called the alternative 5' splice site. Here it should be actually splicing here. Instead of that it is splicing here. That is referred to as alternative 3' splice site. Okay, so such differences, changes in the way RNA is edited can result in many different proteins being produced from the same basic pre-mRNA. Okay, so in addition to this, you should also realize that in eukaryotic systems, uh, you also have what are called as the mRNA, which contains untranslated regions at this end as well as this end. Okay, so the main protein is actually being coded for by exon 1, 2, and 3. This cap, this tail, and this region which are untranslated will never become a protein. Okay. So here is a more fancy picture. So this is the main protein coding sequence. Then you have untranslated region here, untranslated region here. What are these regions? They help the ribosome bind, okay, when translation is occurring. Then you have this cap, which is shown here, which we talked about. Then you have a poly A tail, which is kind of shown here, and that is 50 to 250. Now in eukaryotes, what actually happens is after this complete editing occurs, this RNA is actually stabilized by interaction with a lot of proteins. And actually, it kind of looks somewhat like this. So you have this 5' cap, this RNA untranslated. This is the main region. This is also untranslated. This is a poly A tail, and there's a whole bunch of proteins which then come together along with this 40S ribosome in preparation for the translation. Okay. So, very elegant mechanisms that we have in order to process what is called as our RNA. Okay. So, to summarize, basically what I meant is when the DNA is read in there is eukaryotes, it is read to give you pre-mRNA. Pre-mRNA is processed by several ways: capping, splicing, poly A tailing, etc., to give you our final mature RNA. And this is what is converted to a protein. Okay, so what we just talked about is basically what happens at the level of mRNA. Okay, which is the RNA which finally is going to end up as a protein. Now, post-transcriptional modifications also occur with respect to the other type of RNA, which is tRNA as well as rRNA. Okay, for example, here is what happens both in prokaryotes as well as eukaryotes. Okay, so please remember I told you post-transcriptional modifications are not something which occur only in eukaryotes, they do occur in prokaryotes also. But what you see is that transcriptional modifications which occur in prokaryotes occur only in the level of tRNA and rRNA and not mRNA. While in eukaryotes, after transcription, modification occurs at the level of tRNA, rRNA, as well as what is called as mRNA. Okay. Now, what are some of the changes that are seen with respect to tRNA, both in prokaryotes and eukaryotes? Okay. So here, some of the things that happen is again removal of sequences from each end and removal of sequences from in between, what is called as kind of splicing that you see in mRNA. Okay, so here is an example. I have a primary transcript. Here is base modification, 5' cleavage, 3' cleavage, CCA addition. So see what has happened. Some sequences from here are removed, some sequences from here are removed, and then CCA is added at the end here. Okay. In addition to that, you get some splicing. Okay, so look at this part here. Part of it is removed to give you a shorter part, which is called as splicing. Okay. Now, in addition to this, other things that happen at the level of tRNA is what is called as base modification. Now, if you remember, when you studied tRNA early on in your 11th and 12th, they talk about tRNA having what is called as clover leaf structure. Okay, clover leaf is this leaf which has got actually three leaves attached to one stalk. Okay, and that is seen in clover plants, and therefore this type of tRNA is called as having a clover leaf structure. Okay. Now, also while studying that, they talk about tRNA having what is called as anticodon loop, then D loop, and then T, psi, C loop, and so on and so forth. Okay, and the reason for that nomenclature is actually some of the bases which are present in those regions. Okay, for example, if you look at this part here, okay, which is called as a D loop. Okay, it is called D loop because it contains dihydrouridine. Okay, and here what happens is uridine is actually converted to dihydrouridine by removal of two protons from the okay, actually the other way around. It is dihydrouridine, where two hydrogens are added across a double bond which is present between positions 5 and 6 of uridine. Okay, I do not know whether you recall the structure of uridine. If you remember uridine, and I'm going to kind of draw it here with this pointer.
Two
This is
keto
a
groups.
two keto groups. So this is nitrogen, then there is a keto group, then there is a nitrogen. Okay, and then there's carbon, carbon, and keto group, and joining to the nitrogen. So one keto group is here, the other keto group is here. There's a nitrogen here, there's a nitrogen here, and there is a double bond here. Okay, remember that 5 and 6 position, and that double bond is reduced. Okay, and that's why it is called as dihydrouridine. Okay, and that is present in this loop. There are 2 or 3 uridines which have been converted by what is called as reduction to dihydrouridine, and that gives this loop the name as a D loop. Okay, here what you find is a loop which is called as a T psi loop, CT psi. Okay, T psi C loop actually it is called. The reason it is called T psi C loop is one, there's a thymidine which is present. Now remember, RNA usually does not contain thymidine. Okay, RNA we say is always A, U, G, and C. Now this is an exception to that rule where you see thymidine in RNA, and that's why it's called a T. Okay, the name for the loop is T, and then psi here is for pseudouridine. Okay, a pseudouridine is where the sugar, rather than being attached to the nitrogen of uracil, is actually attached to the 5' carbon of uracil, because it's a very unusual connection of sugar to the base. Normally, if you remember, in all the nucleosides and nucleotides we have seen, the sugar is always attached to the nitrogen. If it's a purine, it is nitrogen number 9. If it's a pyridine, it's nitrogen number 1. In this case, the sugar is actually attached to a carbon. And which carbon? Carbon number 5, which is one of the 2 carbons on the right, which forms actually a double bond. Okay, and that nucleotide is referred to as, I should say, nucleoside is referred to as pseudouridine. Okay, and that is present in this loop. Okay, that's why it is called as T psi, and then always there's a C after that. That's why it's called a T psi C loop. Okay. Then the other thing that you find is a lot of inosines. See this I here in this anticodon loop. Okay, and this I results from deamination of adenine. I stands for inosine. Okay. Now, a little later, no, when we'll be looking at translation, we'll be talking about something called as wobble hypothesis. Okay, anyone heard of this before? Wobble hypothesis.
Yes.
Yes, sir.
Yes,
And
sir.
and in what relation have you heard that wobble hypothesis?
So the last last days of the codon can be variable.
Variable, correct. Okay, and even though it is variable, it is read by the tRNA in the same way. Okay. Now the reason for that is actually the presence of inosine in the tRNA. Okay, and usually all those tRNAs which tend to carry amino acids which have got multiple codons tend to have I here as part of the anticodon loop. Okay, we'll see that a little later. Okay, and that I is present here is a result of deamination. Okay, in addition to that, you also find in this tRNA a lot of places where methylation occurs on adenine or guanine on the nitrogens. Okay, so you get methyladenine and methyl guanine. So all of these modifications like methylation, reduction, formation of pseudouridine, deaminations, they all occur after transcription. So they are referred to as base modifications after transcription of tRNA. Okay, and this is seen both in prokaryotes as well as eukaryotes. Okay, so sometimes, okay, in 11th and 12th, when they teach you transcription for the first time, they say post transcription modification occurs only in eukaryotes, does it occur in prokaryotes? Actually that is wrong. It actually occurs both in prokaryotes as well as eukaryotes, especially when it comes to tRNA and rRNA. However, when it comes to mRNA, that statement is actually true, that in prokaryotes, rarely do you see any PTM in case of mRNA, but you definitely see post-transcriptional modifications of mRNA in eukaryotic systems. Okay? Now, when it comes to ribosomal RNA also, you find that when your DNA is read to give you ribosomal RNA, okay, and this is an example of eukaryotic ribosomal RNA, what you produce is a huge mRNA, I should say not mRNA, a huge RNA from where bits and pieces are chopped off. Okay, so you see here this part is cut out, this part is cut out, and this part is cut out to finally give you what is called as 28S RNA, 5.8S RNA, and 18S RNA. Okay, and this is occurring after transcription. So this is like splicing that you have seen before. Okay, now okay, this arrows which I have shown here are all places where methyl groups are added. Okay, so two things are happening. One, methylation is occurring, and all these red arrows you see are where methyls are added. And the second is also cleavage is occurring, where you get cut out pieces. Okay, now this also occurs in prokaryotes. Okay, so in prokaryotes, again you make a huge RNA from which small pieces are cut out to give you what is called a 16S RNA, 23S RNA, 5S RNA. Okay, and remember these RNAs along with proteins will in the end give you what is called as 30S ribosome, 50S ribosome, and so on and so forth. Okay, so here what I just showed in the previous slide I've shown again to show you that both in prokaryotes and eukaryotes rRNA is transcriptionally modified. I should say post-transcriptionally modified to give you what are called as a mature ribosomal RNAs, which finally become further modified by addition of proteins to give you ribosomes. Okay. Now, we refer to some drugs which affect the process of transcription. Okay, we talked about amanitin last time, but there are here two other drugs which are also inhibitors of what you call as formation of RNA polymerase. Formation of RNA by inhibition of RNA polymerase, actinomycin and rifampicin. Okay, now actinomycin D actually is used for treating cancer. Okay, and the reason it is used for treating cancer is because it's a very good intercalating agent. That means it actually binds to double-stranded DNA and doesn't allow the double-stranded DNA to separate. But one of the other effects that actinomycin also has is that it after binding to the DNA template, in addition to not allowing the DNA to separate, when RNA polymerase comes, because the DNA cannot separate, it actually blocks the movement of RNA polymerase. The other is rifampicin, which is used to treat TB, okay, and it binds to beta subunit of prokaryotic RNA polymerase. That means it binds to the bacterial RNA polymerase of mycobacteria, and therefore it inhibits the growth of mycobacteria. Okay, and as I said, amanitin is a toxin. It actually binds to eukaryotic RNA polymerase and it inhibits protein mRNA synthesis. Okay, so these are three drugs which affect transcription. This affects transcription in humans. This also affects transcription in humans, and rifampicin affects transcription in bacteria. Okay, more specifically, mycobacteria. Okay. Now the other aspect is that last time somebody was asking about why do we have, in terms of promoters and enhancers and signal sequences, why not just have it very simple, that you have a piece of DNA which is directly gives you the RNA and you take it from there. Okay, part of the reason for that is so that you can control and regulate the way transcription occurs. Okay, so if you were to look at all the genes that are present in a biological system and the way they get expressed, you'll find that you can actually classify all the genes based on the way they get expressed into two groups. Okay, one which are called as constitutive genes, which actually get transcribed all the time at a background level, and because of that you will find that they also produce the proteins all the time at a background level. Okay, now these genes and the proteins that are produced by these genes are actually required in general for your keeping your cell functioning. Okay, and they are referred to as housekeeping genes and housekeeping proteins. Okay, an example of that would be, some of the enzymes which are involved in, say, glycolysis, TCA cycle, etc., which are processes which occur in almost all the cells in order to generate energy for the cell to survive. So these processes are not something which do not occur, and then when you need they will occur and then they stop. They keep on occurring in the background all the time. Okay. So such genes and their expression, which occurs constantly at a baseline level, are referred to as constitutive genes and constitutive proteins. As opposed to that, you can have also genes and the proteins which are produced from that genes being expressed only when necessary. Okay, and these are referred to as inducible or adaptive genes or inducible or adaptive proteins. Okay, a nice example which you have also heard of maybe in your 11th and 12th, as they are saying, is that you must have heard of how some bacteria which normally grow on glucose are also able to grow when you give them lactose. And the way
So
they're
lack
able to,
of peron.
yes, the lac operon, correct. So the way they're able to do that is because now they sense that there is no glucose, but there is lactose in the medium. They synthesize all the proteins which are necessary to break down lactose to give galactose and glucose on a need to basis. Okay, so they will do this till lactose is there, and once lactose gets over, they will just simply switch off and not express those genes. So such type of genes which are made when necessary and then stopped and they don't occur normally in the cell are referred to as adaptive or inducible genes. Okay, so these are two groups. Okay, and in addition to this, there are other ways, other situations also. But let us look at some of those things. Okay, now the way to regulate transcription, okay, and is in one way is what they call as alternate sigma factors usage, okay. So if you remember what was the sigma factor when we looked at RNA polymerase, we said the sigma factor is the one which is actually required for initiation. Okay, so it binds to the promoter, it binds to what is called as the Primnov box, no, minus 10 region and minus 35 region in case of prokaryotes, and the minus 30 and the minus 75 region in case of eukaryotes. And the sigma factor is there along with the RNA polymerase till the RNA polymerase starts the transcription. After that, it simply falls off. Okay. Now, this sigma factor can be used to control what is called as which genes get expressed. Okay. So, here is an example of how what you call as sigma factor 70, sigma factor 32, sigma factor 60, they all play a role in expressing different types of genes. Okay, these are what are called as vegetative principal genes. Then the other is they are induced when when the bacteria are subject to heat shock. The third gets induced when there is a low amount of nitrogen in the medium. Okay, so under these situations, different sets of proteins are produced, and the way they do is by using different sigma factors, which then start initiation at different points by reading different types of promoters. Okay, see this different types of promoter: CTGGNA, CNCTTGA, TTGA, GACA. This is what we saw as a constitutive one. Okay. Now, the other way to kind of have a handle on which genes get expressed and which genes don't get expressed is by again we say positive regulation. Okay, this is what we saw. These promoter sequences and activator sequences are where proteins bind, and when they are bound, RNA polymerase is able to bind better and initiate transcription in a much better fashion. Okay, so like this activator improves the ability of RNA polymerase to bind and initiate transcriptor at what is normally a weak promoter. So this is allowing that promoter to become a strong promoter. And the reverse can also occur, what is called as negative regulation, what you call as repression. So what happens here? A protein factor blocks the ability of RNA polymerase to bind and initiate transcription at what is normally a strong promoter. So what happens here is, normally the gene is read very well, but when this repressor is present, I inhibit the transcription of that gene and slow down everything. Okay. So these are all different ways you regulate transcription. The one you have kind of heard of before is what is called as operons. Okay, in operons, what happens is a whole set of genes are produced when needed, all at one shot, and then when there's not needed, all of them are shut down. Okay, one classic example, as you already said, is a lac operon. Okay, so in lac operon, what happens is uh, the minute the bacteria senses that in the medium there is lactose and there is no glucose, it immediately starts producing all the proteins that are necessary to utilize lactose which is there in the medium. Okay, and the reason for this type of setup is that in bacterial systems, environmental changes rapidly, and many times if the bacteria is unable to respond to this change in environment, it will not survive. So what the bacteria does in many cases is keeps many enzymes which are required to do a certain function organized on its gene as part of an operon. That means what? Either they will all get expressed or none of them will get expressed. Okay, and the reason for this is I can then control how much enzyme I make. Okay, and control when I should make it. Because if you remember, making enzymes is not cheap, because to do translation and to join amino acids together to give you proteins costs a lot of ATP, as you will see when you do translation. So what the bacteria does is makes it only when it is required. Other than that, it doesn't make it. But the way it sets it up is that the minute it senses that the enzyme is needed, it will trigger the synthesis through what are called as these activator proteins. Okay, so see what happens in case of lac operon. Here is the operator and there is a promoter. These are all sequences where this lactose or uh, proteins which are activated when lactose is present in the medium are going to bind and regulate the expression of three genes. In this case, all these three genes give different proteins and all of them are actually required for utilization of lactose. Okay, so the idea is I can regulate a single promoter and I can coordinate the expression of three genes in one shot. Okay, rather than having separate operator and promoter for each and every gene. Okay, okay. Now in eukaryotes, we generally do not have to tend to have this system. Okay, and maybe the difference is that being more evolved organisms, we rarely find ourselves in a situation where we need to adapt so quickly in order to survive. Okay, so over the years, as we become more and more complex, we have gone away from this type of system. Okay, and even if they are there, they occur, they are few and far between. But in bacteria, this is very, very common. Many genes which are lined up one after the other, and they'll all get expressed, or none of them will get expressed in response to a certain stimulus. Okay. So what I'm going to do is actually kind of stop here because this is where I'm going to end my transcription. So what I've done is basically ended transcription, talking a little bit about how transcription can be regulated either what is called as negatively, positively, or by using sigma factors of different types in response to different situations. Okay, and the fact that some genes are constitutive and some genes are inducible. Okay, so I'm going to stop here, and when we meet next time, we'll begin the last process in this life process of DNA, which is translation. Okay, so I'm going to stop sharing here and see if there are any questions. Answer those questions, and then we'll call it a day for today. Okay, so let me see here. Okay, chat box. Huh. Why there was a need to adopt RNA editing? Okay. In splicing, there are different enzymes for alternative splicing. Yeah, okay. Many times alternative splicing is controlled at the level of the tissues uh, and in the level of what is the cellular status. Okay. And yeah, it is entirely possible that different enzymes may be used for alternative splicing. Okay, why do we need RNA editing? It is one other aspect which is a way to do more with less DNA. Okay, so see all of these things, no, alternative splicing, RNA editing, these are actually probably all remnants from the fact that we have probably evolved from prokaryotic systems. Okay, now, since we have all evolved from prokaryotic to eukaryotic, there are some things which are part of the prokaryotic systems which you still carry forward. Okay, and a part of that is this the idea of getting more from less DNA. Okay, the way I do it, get more from less DNA, is I take DNA, I convert it to RNA, and then I use that RNA differently so that I can get different protein. Okay, so and that is what bacteria tend to do because their genomes are smaller. So a part of that is still carried forward by us, even though we have evolved to be a little more complex. Some signs of our evolution from prokaryotes are still there. Okay. Now again, Aditi is asking, why would we need a part of intron and why would we remove part of exon, and that we have different types of splicing, and how is it actually done? Again, many of these, okay, see, the way splicing occurs is of two types. One, what is called as self-splicing. Self-splicing is where the RNA itself has the ability to cut itself out. And the other type of splicing is called as assisted splicing, where you actually have enzymes which help you with splicing, and they are actually proteins. They are not RNA, but they will splice RNA for you. Okay. Now, the question, why do we need part of intron? Why would we remove part of exon and keep it? These are all probably to try and get different types of proteins using the same set of genes. And again, as I said, much of this is seen in prokaryotic systems because they tend to use their genome much more wisely than us because they don't have that much genome. Okay, but you also find, as I said, some remnants of this also in eukaryotes because we have evolved from prokaryotic systems. Okay, and how is it actually done? Yeah, different enzymes recognize different sequences, and this will be very, very specific sequences where you can do this cutting to get this very alternate type of transcriptional modification of RNA. How is base modification done in tRNA help in its function? The changes are kind of unheard. No, they are not actually unheard of. Okay, methylation of DNA, methylation of RNA, acetylation of DNA are all known, very, very well known changes. In fact, if Aditya, have you heard the term epigenetics by any chance? Aditya?
Sorry, epi?
Epigenetics. Have you heard this term,
No, sir.
epigenetics? Okay.
No.
After this lecture is over, no, just go to Google and type epigenetics, okay, and you'll be able to read something very interesting. Now, what is it? I'll tell you what is it very briefly. Epigenetic changes are changes which occur at the level of DNA, which in many cases are transient changes. Okay, that means this is going to occur for a short while, and then in many cases this will never be passed on to the progeny. Okay, and what actually happens in epigenetic changes is that specific sequences or bases in your DNA gets methylated, and because they are methylated, what happens is there is a change in the level of expression of certain genes. Sometimes certain genes do not get expressed at all because they are methylated. Sometimes they get expressed 5 or 6-fold better because they are methylated. And because of this, what happens is, even without changing the DNA sequence, okay, even without changing the DNA sequence, you are actually changing the way the DNA is functioning. Okay, and such changes are referred to as epigenetic changes. Now, very interestingly, they have also found that some patterns of this methylation, which is called as epigenetic changes, are appear to be also inheritable. That means this pattern of methylation is somehow passed on to the progeny also, because the progeny also show this type of methylation patterns. Okay, so as I said, these changes, although it might appear to you are unheard, they are fairly well known, and they've been well known for a long time actually. Okay, so take a look at what these epigenetics are, and maybe that will answer a little bit about this methylation. Okay, so yeah, uh, phenotypic changes without changing the DNA sequence is what we mean epigenetics. No, phenotypic changes without changes in DNA sequence is usually only at the level of what you call as changes in expression of DNA. Okay, for example, if I carry a piece of DNA, okay, and and the way the DNA is read and the protein that I get, that gives me a certain phenotype. Okay, now, if let us say I've got a gene which gives my eyes blue color, okay, so I can look at my eyes and say, okay, the reason I've got blue color is because my DNA has sequences of this type. Certain other person might have what you call as brown color eyes, and you can say that this brown coloration of the eyes is because the DNA sequence is this. Okay, so now what we are talking about is two different DNA sequences, which when expressed give two different colors to the eye, and these are called as two different phenotypic traits. Okay, now in this case, what is happening is you will probably not be able to change the color of the eye from blue to yellow, but what you might be able to do is making it much more bluer or light bluer. Okay, because the gene per se is the same, but because of this methylation pattern, either the expression starts to be more or expression starts to be less. The protein which is produced is probably still the same. Okay, so such variations are actually, in a sense, phenotypic changes but at the level of changes in expression. Okay, and that is what is called as epigenetics. Now, how can one identify between exons and introns? Okay, there are some sequences which are present in certain types of introns, which which gives you a signal that these are introns. Okay, but such sequence specificity is not there in every type of introns. Okay, so some introns do not have those features which tell them, which you can look at and say, okay, these are introns. Okay, in certain cases it's very difficult to identify, but in certain cases, for example, especially in self-splicing introns, there are certain sequences which are hallmarks of these types of introns, and there you can recognize them. Okay. So, did I answer most of the questions?
Sir, actually, I wanted to ask
Huh.
sir,
Yeah.
we have all these changes in tRNA, so it kinds of help tRNA in its function or something like that.
Mm-hmm. tRNA, right?
Yes.
Okay.
So all these changes occur. So how does it help actually in its function? I wanted to ask that actually.
Okay. Okay. Now, see the difference is in case of tRNA, uh, some of the changes that occur actually are part of making the tRNA more and more specific in terms of what amino acid it is going to load at the end. Okay, and what anticodon it is going to recognize. Okay, uh, so actually speaking, if you look very carefully, it is not that we have only one sequence for tRNA, and based on the modification, then you are able to load different amino acids and have different anticodons. That is incorrect. Actually, we have several genes which code for different RNAs which are actually going to carry different amino acids. Okay, but even then, before they can load the amino acid efficiently, before they can bind to the anticodon loop efficiently, the tRNAs need to be modified. Okay, so some of these modifications are actually so that they are able to bind to the target amino acid better. They are able to bind to the anticodon which is present on the mRNA better, and that they are now recognized also by ribosomes as the correct RNA which is come and sat in the ribosome before the ribosome will form the peptide bond. Okay, so some of these modifications are for those purposes, the stability of the RNA and the functioning of the RNA. Okay, but again, please remember, we do not make only one tRNA and then based on the modification it will take different amino acids. We actually make lot of different RNAs for different amino acids, and each of them is subject to different types of modification. Okay, does that answer your question?
So, but all that methyl groups addition and hydrogen groups removing and all
Correct,
that
correct.
stuff, won't it kind of, affect the stability and the structure, like make it more or less stable if atoms are
Yeah,
added and removed?
yeah.
Won't it
All of
kind of
these,
affect?
it does, it does. Every change you make to a tRNA is actually going to change the property of the tRNA. Okay, because some changes might appear very small, like putting two hydrogens, but some changes are very big. For example, if you splice out a whole section and all, okay, and again, when you convert, say, cytosine to uracil, uh, or make pseudouridine, okay, these changes are also very big changes. Or cutting off a piece from the 5' end and 3' end and adding CCA, that is also going to change the function and stability of the tRNA. But I feel that some of these things that keep occurring in your body are all again remnants of your evolution from bacterial systems where all of these things used to happen. Okay. Now, nobody actually knows the reason for every change. The only thing they know is they observe all of these things happening when the tRNA matures and before it is able to load the amino acid. Okay, and I also feel that there is a lot of unknowns here, but what we are actually seeing as far as all of these modifications that I'm talking about is a lot of things which are being left over from the fact that we have evolved from bacterial systems where these things happen all the time. Okay.
Yes, sir. Thank
Yeah.
you, sir.
Yeah, yeah. Okay. So everyone fine? Can I stop at this point? So let me quickly do one thing. I'll download the attendance list and shall I stop recording if there are no more questions? Any
Yes,
more
sir.
questions? Okay.
Yes, sir.
Okay. I'm stopping my recording.